Very good morning. Today we'll be discussing laboratory animals, which is a very important topic in in pharmacology in both theory and practicals. And we know how much important laboratory animals because due to similarity of the laboratory animals, we used to recommend a dose and drugs, and even though we used to screen the drugs, whether it is a new or existing drug in the human. So let's have a look which are there inside the screen. The first animal we have is the rat. The scientific name of the rat is Rattus norvegius. The recommended weight is 200 grams. The here, so what is recommended weight? So it is a weight in which we can carry out a pharmacological experiment. So if we talk about the range of weight, then we consider 180 to 220 gram. So it is the ideal range in which we can carry out the animal experimentations. Due to the color of the rat, we call albino rat. Albino means color of the coat, and it is most commonest laboratory animal, and it is most suitable animals for experimental due to its small size and shows great sensitivity towards most of the drugs. And it is most standardized of all the laboratory animals because whenever we see the organ or pathophysiology of rat, we found similarity in the humans. And we found maximum similarity in case of rat and different organs or the cells that we have used for different purposes in pharmacology. Furthermore, we can breed or obtain pure and uniform strain from the rat. Due to its small weight and the captivity is easier. So there are two strains the mostly we use in pharmacological experiment. One is Wister rat and the second one is Proudhon rat. So first we understood that normal or the known name is rat due to color of its color or the fur it called as albino rat or due to white in color we call it albino rat. And further we have divided into two. One is Wister rat, another one is Proudhon rat. And we can say it is why we are classifying. It is based on its morphologically and also based on its characteristics. So Wister rats are very quiet, moderately prolific, strain, rather resistant to infection. Here, the most of the Wister rats are very quiet. They used to stay calm always, and their growth rate is moderate and highly resistant to infection. So most of the time, the scientists choose the Wister rat. If we see the morphology or anatomically, we see that head is wider in case of male and ears are long. And the tail length is always less than the body length. So this is all about the Wister rat. So when we're talking about the Sprouse rat, so it is rapidly growing and more prolific than Wister rat. So there are vast changes or significant changes between the Wister rat and the Sprouse whereas Wister rats are quiet, but Sprouse is not as quiet as compared to Wister. If we see the growth rate, Wister rats are moderately prolific, whereas Sprouse rats are rapidly growing. If we talk about the resistance, Wister rat having the high resistance. In case of Sprouse it has less. Wister rat has a longer and narrower head and longer tail, and which is just opposite of the Wister rat. And it has less resistance to the infection and mostly respiratory disease. So most of the time we are not choosing the Sprouse rat for respiratory disease or lungs disease. So now we'll move towards the anatomical significance of rat. The first anatomical significance we found that rodents, which includes the rat, mice, hamster, that doesn't have vomiting center. As it doesn't have vomiting center, we cannot carry out any kind of emetic study or any kind of anti-emetic study. So we must need to take another alternative animal for this study. Second, rats do not have tonsils or gallbladder. So again, the study which is related to the tonsil or gallbladder we cannot carry out. Third is they have extremely diffuse pancreas. So here what does it mean extremely diffuse pancreas? If we visualize the liver or if we visualize the stomach, it has a particular shape, particular weight. But in case of pancreas, here it is diffuse, so we may not get that specific shape pancreas, but it is present and we can also carry out the experiment. Next is the rat, being omnivorous, resemble to man nutritionally. As rat omnivores, their style of having food or their food or nutrients what they require, it is resemble to human. As their food are omnivorous in nature, so there are many pathophysiology or biochemicals parameters are similar to the human, and we can carry out those tests. As like human, rat also shows the menstruation cycle, which we call estrus cycle. So it means that we can carry out the reproductive studies rats. Next, rat as an experimental animal. So rats are found to be very sturdy to withstand long period of experimentation under anesthesia. So by using rat, we can carry out long-standing experiments with or where there is a need of sacrifications also we can go for, or where there is a need of recovery also we can carry out. For example, some studies where we are going to collect the blood, studies where we are going to collect the blood sample or any kind of urinary sample we can collect. Again, there are some studies are 21 days, there are some studies are 28 days, but there are some studies where we need to isolate in ileum or heart. So based on different kind of experiment, we can use the rat, whether it is a short period of time or whether it is a long period of time, including toxicity study. Second, as an experimental animals, rats are particularly suitable for testing psychopharmacological agents. So rats are also being used for screening the drugs which focusing on mind or state of minds. So rats are also preferred for assay of different hormones, and we know how hormones are important or required in our body. They are local hormones or general hormones, but they are always helps or maintain the body homeostasis or our endocrine and exocrine glands. So these animals are also employed for the study of drugs on the blood pressure in acute and chronic or surviving experiments. So it helps us to screen the drugs in different conditions. Again, the rats are also prepared for toxicity study, whether it is a acute, whether it is a chronic, whether it is a repeated, or whether it is a long or subchronic. And even though whenever it is about the selection of the organ, we can go for uterus, stomach, colon of the rats, which are routinely employed for the study of drug. So rats are also employed for the study of analgesic drugs by the method radiant heat application on the tail, or there are many. For example, Addis phosphate, von Freisis. So there are many ways that we are going to study the rats and their properties on pain. So it is again useful for the physiology of liver. So we can study the drugs, how it acts on the physiology of the liver. Again, rats are very suitable for testing the drugs for teratogenicity and carcinogenicity. And we know teratogenicity is a very important topic we must need to discuss, and which is nothing but, so when any drugs or any that crosses the placental barrier, we must call them teratogen, and there is a chance that they will develop a phenomenon we call as teratogenicity. And we found in mostly in the pregnant woman, so we must need to careful, and before it goes to the human, we always screen in the rat. Again, carcinogenicity. Now, due to the our advancement of the age or advancement of the days or the lifestyle, there are many drugs or radiation that we always expose. There are many drugs that we are consuming, or there are many articles that we are consuming. So we must need to check all everything drugs article that are directly or indirectly applied in our human life or we are ingested, so that it will not going to develop the carcinogenicity being a carcinogen. At the last, rates are also employed in the biopsy and preclinical trials. And biopsy is not new for us, and we know it is estimation of concentration, potency, and efficacy of an unknown drugs on experimental lab animals by using the biomedical devices. So where most of the drugs are the concentration of the drugs are unknown, and we are going to identify, are there other methods other than biopsy which are chemical radioimmunoassay. And next is preclinical trials. So preclinical trials is very important phase in development of new drugs. So any drug before it goes to the clinical trials, the preclinical trials is to be done, and preclinical trials, it consists of in silico, in vitro, and in vivo. And this completes today's class, and we will continue in the next class. Thank you.